Alright, so today we have the protein synthesis project, except for we're going through everything. So first off, we have our DNA strand. Now, the top strand, TAC, TTG, TTC, ATG, GGA, and ACT, well, that's our original DNA sequence. The stuff below it is what our original DNA sequence pairs with. Now, as for DNA is always made of a sugar phosphate background, and all the nitrogen bases, thymine, guanine, adenine, cytosine, the TACs and Gs, those are all called nitrogen bases. And all the nitrogen bases are on that sugar part of the backbone. And as you can see, the sugars and phosphates, they repeat back and forth. Now, if you do notice, you can see that T pairs with A and G pairs with C. And it will always be like that in every DNA sequence. So you always find DNA in your nucleus. This is where you find everything. In the nucleus, well, let's see, inside the nucleus there's a nucleolus, inside and outside the nucleolus in the regular nucleus, that's where you find all your DNA and stuff. And then on the outside of the nucleus, you'll find nuclear pores, and yeah, so that's fun. Okay, so, but we're talking about protein synthesis, which is making proteins, right? So, in order to make proteins, we have to have the DNA. See, this is our DNA strand and the double helix. Well, that needs to split apart, and we have to make something called mRNA. So, as you can see, DNA splits apart, and then all these little things on the outside, these mRNA things, well, they'll come in and they'll pair with all of these other ones. Now remember, since it's mRNA, so RNA, so the R in mRNA is a different sugar, so it has a different sugar. Because of this, it also has a new chemical, and that would be the U in your cell. And U really just replaces the T. So T replaces U and stuff. So anyway, all these mRNA nucleotides are called, they come in and they pair with all of these things, with our original DNA strand. Right. So then if you come down here, we already explained the sugar is different, and the U, which replaces the T and regular DNA, not RNA. And this is our DNA sequence. So now we have TAC, TTG, TTC, ATG, and GGA, and ACD. That's our original DNA sequence. And that now pairs with, and this would be our mRNA sequence, AUG, AAC, AAG, UAC, CCU, and UJA. That's our mRNA sequence. And all this happens in the nucleus, okay? So, now, all of these wonderful mRNAs program for these 5 amino acids, MAT, ASN, LIS, and TYR, and PRO. And that is our amino acid sequence, and that is how we make a protein. But how do we get the MNR sequence to program for these things? Well, in order for that to happen, we have to go back to over here. So the reason why DNA needs to split and make an mRNA is because DNA, protein synthesis, happens in these little things called ribosomes, all these little circles throughout here. And DNA cannot leave the nucleus. So therefore, we have to have all of our mRNA get out to go make ribosomes, right? So the mRNA leaves and travels into the cytoplasm. And in the cytoplasm, they travel to ribosomes. And once they travel to ribosomes, we get this. Now, mRNA always has groups of 3. AUG, AAC, AAG, and those are called codons. And these codons, 3 codons, are 3 nitrogen bases make 1 codon, and 1 codon programs for one amino acid or a stop to that amino acid. So in order for these codons to figure out which is the right amino acid, they have something called tRNA with an anticodon, which is basically just the opposite of the mRNA come in with, and a tRNA is a transport RNA with, and it has all of our proteins, as you can see, MAT, ASN, LIS, TAYR, PRO are amino acids, and they'll come in. So the anticodons pair with the codons, and then if it pairs correctly, then it drops off our amino acid, and the tRNA leaves to go pick up more amino acids, and the amino acid stays and pairs to the next one. See, another codon paired, and that one will leave, and then repeats all the way till you get one of these stop ones, the ACU, which pairs with the UGA, and then it'll stop. And then finally you get these 5 proteins, and look at that, perfect 5 proteins. And if you look, remember, these are the mRNA and the tRNA are opposites, so UAC, UUG, UUC, AUG, GGA, ACU, that's our tRNA code. And those, if you pair them up, would be opposites of AUG, AAC, AAG, UAC, CCU, and UGA. And remember, mRNA codes for your amino acids, and the transports have those anticodons. And as you can see, since it's still RNA, we have the U, and we're not using these T's. And one other important note, you can see it's all happening in the ribosome. See the shape right here? That's our ribosome. That's important, and that's where this all happens. So all of our little amino acids, they're all in the cytoplasm stuff, and the messenger RNA is in the cytoplasm and in the ribosomes, as you can see by this diagram. Now, sometimes we have mutated DNA sequences. So basically, our one mutation is called a point mutation. So as you can see, we go through our original DNA sequence, everything's normal, and then we have a mutated sequence. And in the mutated sequence, we have 4 normal amino acids, it's the same thing, but then we have this A changes into a G. Now, this might not seem, and then you program for that DNA mutated sequence, goes through that, and you see now it's CCC. And then it goes through again, and we get GGG is our new tRNA, which our tRNA will now bring it'll bring the same thing. In this point mutation, there is no change because it programs for the pro amino acid, and we still have the stop, so there is no change. It's called a silent mutation. But say, what if this right here was actually AGA, which would become the messenger RNA would be UCU, and that would actually program for SIR, and so an amino acid is changed. So that could happen sometimes. Another type of mutation is the deletion frameshift mutation. So as you can see, and we have our original DNA sequence, and then in the deleted DNA sequence, we delete a T. And so all these nitrogen bases have to move over one. So 1, 2, 5, 8, 11, 13 amino acids, 13 nitrogen bases are now out of place. So we have a new MNR sequence, as you can see, a new tRNA sequence as seen here, and this changes 4 amino acids. Now we have THR instead of ASN, we have SER instead of LIS, we have THR instead of TYR, and we have LEU instead of pro. So 4 amino acids change, and we no longer have stop. We no longer have a stop command. So it's all messed up. That protein is all messed up. In the same way, we also have the insertion frame shift. The insertion frame shift, we have four of the original DNA sequence stays the same, but now in the next one, the inserted DNA, we added an A in this one, the third letter. So since we added an A, we have a T left on the end. So, and this amino acid has now changed. This amino acid, while it still having something inserted to it, is the same. So this group doesn't change, but this group does. So then we have our mRNA sequence, and you can see this one's a little bit different. And we have our tRNA sequence, and same thing, just follow the base pairing rules to get those. But let's look at what happens in that insertion frame shift thing. So now we have MET, ASN, list, tire, and pro, which, you know what, we have all the same things over here, but we no longer have a stop protein. Instead of that stop protein, we have a LU amino acid program. So there's no stop message. So now this protein will also be pretty messed up. And that's basically protein synthesis. This stuff all goes through this stuff right here. This is called our transcription, is this part. And once you go to transcription, then you translate it. And then after that, you get your protein. And every once in a while, you have to watch out for mutations, which will make a protein that your body doesn't want. And this all starts in the nucleus, then will all go into your ribosomes. Okay, I think that's it. I don't really know.